Dear listeners,
Hold on.
recently, Chinese scientists have achieved an unprecedented technological breakthrough, reversing adult blood cells into infant-stage stem cells for organ regeneration and anti-aging. This is, without doubt, an exciting medical revolution, with the potential to solve numerous long-standing health challenges. Yet, skepticism remains. Whether this technology can ultimately reach the market still depends on rigorous validation and certification. Today, we begin with the so-called human cell factory and dive into the scientific logic, ethical debates, and the transformative future this innovation might bring.
Welcome to The Debate. Today, we're diving into a really fascinating report. It describes what many are calling a potential paradigm shift in biomedical science. The successful reported reverse engineering of adult blood cells into, infant-stage stem cells by researchers in China. Now, this process supposedly harnesses what the source material calls unlimited potential to generate new tissue. And naturally, this immediately raises some pretty profound questions about the limits of human biology and the very nature of aging itself. So our central question today is this. Is the evidence presented in the material, specifically thinking about that highly successful 33-month treatment of diabetic patients and the massive commitment shown by building the Harbin cell factory, is that evidence sufficient to view this technology as a definitive near-term revolution? One capable of ensuring on-demand organ regeneration and maybe even fundamentally challenging the long-held notion that aging to be irreversible?
That framing, I think, captures the core tension here perfectly. The reported success is, unquestionably impressive. No doubt about that. But the claims being derived from it, a fundamental revolution in human health, the immediate end to donor reliance, even aging reversal, those are, frankly, staggering extrapolations.
Right. And I'll be taking the position that the combination of factors, the clinical success we've seen, the potential guaranteed solution to immune rejection, and that institutional commitment shown by the factory, I believe this confirms we are indeed witnessing a revolutionary shift, one that could fundamentally redefine human health.
And I'll be arguing that while the initial success is profoundly important, yes, the sweeping claims, particularly about aging reversal and systemic, immediate organ regeneration, are currently, future projections. They're built upon impressive, certainly, but ultimately highly specific and therefore functionally limited initial evidence.
Okay, so my core thesis really rests on the breakthrough's apparent ability to solve the main barrier in transplantation medicine, immune rejection. By using, as the material puts it, the patient's own drop of blood, researchers can reportedly cultivate perfectly matched organs and tissue. This guarantees no rejection reaction. Now, that's not just an incremental improvement, that's a foundational change. And we have proof, right? We have the success story where diabetic patients used these regenerated islet cells and achieved continuous insulin independence for 33 months. Their blood sugar levels actually normalized. This proves that the resulting cells aren't just viable, they're fully functional over a pretty substantial period. The implications, I think, are clear, just as the material states. Complex organs like the liver or kidney could potentially be created on demand that would permanently end our reliance on donor pools.
And furthermore, and this is, I think, critical to the scope of the revolution, this process of cellular reversal directly challenges the traditional notion that aging is irreversible. It offers a whole new possibility for lifespan extension. The scientific mechanism validated by this clinical success, it really seems to support these, ambitious claims.
That is a compelling argument, I grant you. But I do believe we must maintain a degree of scientific discipline when we move from, essentially, a proof of concept to declaring a revolution. While the technology shows undoubtedly immense potential, particularly in treating type 1 diabetes, the broader claims of redefining human health and achieving immediate systemic reversals, these feel like premature generalizations. They're based on a very specific, very localized success. To elaborate maybe a bit on the mechanism, this reported reversal of adult blood cells back to an infant stage state Well, it's likely related to, or perhaps an advanced form of, cellular reprogramming, something akin to the induced pluripotent stem cell, the iPSC technology that, we've been exploring for years now. However, if this new technique genuinely offers superior stability or efficiency or maybe epigenetic purity, which that infant stage designation seems to imply, then yes, that is a major biological leap. Still, the demonstrated clinical success is restricted solely to islet cells, and only over 33 months so far. Now, islet cells, though functionally complex, they represent an endocrine tissue, essentially a cellular aggregate, not a large, structurally complex, vascularized organ like a liver or a kidney. So to claim that this success guarantees the on-demand creation of a liver or a kidney, well, we'd need far more evidence, wouldn't we? I just can't accept that a successful tissue replacement automatically guarantees a massive, generalized outcome like instant organ regeneration simply because the rejection problem might be solved. And the conceptual distance between repairing specialized tissue and challenging the irreversibility of aging, that's just too vast a leap to take based on this single data point, in my view.
Okay, let's delve deeper into that specific point then. The weight of the evidence. You frame the 33-month success as specific and limited. Whereas I see it as validation of the foundational principle, the most challenging biological principle, really. So is 33 months of insulin independence sufficient to justify claims about systemic applications like liver and kidney regeneration? I argue yes, because that 33-month functional result confirms the viability of the core mechanism, the successful, apparently stable conversion of adult blood cells into functionally mature, non-rejecting cells. This validation, the pluripotent nature and stability of these infant-stage stem cells, that's arguably the most difficult hurdle in regenerative medicine.
Once that core principle is proven that you can create perfectly matched, functional, disease-free tissue that remains stable long-term, then the application to organs with greater structural complexity, say the kidney or the liver, it becomes more of a solvable engineering problem. The cell-fate pathway is confirmed, the biological foundation seems secure. Arguing that structural complexity is the primary barrier now sort of ignores the fact that modern bioengineering is rapidly mastering issues like scaffolding and vascularization, often using things like bioprinters. The cellular breakthrough is the ultimate bottleneck, wouldn't you agree? And the materials suggest that bottleneck has been overcome.
I'm not quite convinced that simply solving the cellular bottleneck makes the structural challenge trivial. I think you might be minimizing the fundamental complexity of a solid organ there. The liver, the kidney, they require incredibly intricate three-dimensional functional architecture. They rely on microcapillaries, supportive stromal networks, a precise hierarchy of organization. That's currently beyond the scope of just growing cells in a dish, even if they are perfectly matched cells. The breakthrough proves tissue regeneration is possible. Yes, I fully concede that. That's huge. But generating a large, viable organ that can actually filter toxins or metabolize nutrients effectively, that requires the cells to either self-organize perfectly or be placed precisely within a complex scaffolding structure that can survive implantation and integrate.
We haven't seen proof of concept for the successful regeneration of an entire complex, vascularized organ from these specific stem cells yet. We really mustn't confuse, generating the building blocks with successfully constructing the entire skyscraper.
Well, while I acknowledge the difficulty of bioscaffolding, the fact that these are described as infant-stage cells, that phrasing itself might be key. That terminology could imply the cells retain a potent ability to organize, not just differentiate. Maybe the innate biological instructions for forming those vascular networks and that stromal architecture are still built into the cellular material itself, just ready to be deployed given the right environment. Perhaps the factory's purpose, as we'll discuss, is precisely to create that optimal environment. The confidence that seems to radiate from the material suggests that the researchers believe these cells, unlike maybe less stable or more mature cell lines, possess the necessary developmental capacity to handle that structural burden.
Okay, that brings us to what I find perhaps the most conceptually challenging claim in the material. challenging the irreversibility of aging. I approach this from a slightly different angle. If the primary achievement here is replacing a specific damaged component, like the failed islet cells in diabetes, then we're fundamentally talking about precision anti-disease medicine, which is tremendous for longevity, yes, but indirectly by preventing death from organ failure. But is replacing one worn-out component truly equivalent to systemic aging reversal? Real lifespan extension, in the broad sense, surely requires halting or reversing the myriad degradations across the entire organism. Neuronal decay, telomere shortening in multiple cell lines, mitochondrial dysfunction across all tissues, you name it. So we need to clarify, I think. Are the researchers simply proposing replacing individual failing parts, thereby extending health span and delaying disease-related death? Which is fantastic, don't get me wrong. Or does the mechanism itself, reversing adult cells back to the infant stage, actually confer a true systemic reversal of biological age that impacts all systems, not just a replacement tissue? Because if it's just component replacement, well, it's not really aging reversal in the way that phrase is typically understood in the field. It's more like magnificent biological maintenance.
Ah, but I think you might be drawing a line there that the technology itself is attempting to erase. The claim that this technology can fundamentally rewind cellular age, that is the core philosophical driver here, isn't it? If old, failing, age-decayed tissue, whether it's an islet cell or heart muscle or kidney nephrons, can be perpetually replaced by biologically young, perfectly matched tissue cultivated from the patient's own resources, well, that is a direct and I'd say profound challenge to the traditional notion of inevitable, accumulating age-related decay. Look, aging kills us primarily through cumulative system failure. If we gain the capacity to replace the most critical failing systems, liver, heart, kidney, endocrine function potentially on demand, we fundamentally alter the shape of the human life curve. We wouldn't just be preventing death from diabetes. We'd be offering a mechanism to circumvent the biological clock that governs organ viability. Now, full systemic rejuvenation might still be years away, granted. But the successful replacement of key age-limited organs with biologically young ones, that opens a new possibility, as the text says, for extended life that was, frankly, science fiction until very recently. So I argue it's not just maintenance, it's scheduled high-level biological renewal.
That is certainly the ambitious goal of much anti-aging research, yes. But the practical hurdles remain enormous. Even if we fully accept the capacity for cellular renewal, the sheer resource demand required for routinely manufacturing bespoke, individualized organs for every patient, potentially across multiple organ systems, it just makes this revolution sound quite distant, not immediate. If the science truly is sound, as you suggest, then the only remaining barrier to widespread revolutionary impact becomes logistics. And that, I think, is precisely why the existence of this Harbin factory needs careful scrutiny.
Exactly. And that logistical challenge is precisely what the material addresses directly, which leads us neatly to our final key point: scalability and immediacy. The construction of the world's first human cell factory in Harbin, which is explicitly intended to drive the technology towards scaled application, well, that's not just an investment, is it? It's a declaration of confidence in immediate real-world implementation. It moves the entire endeavor squarely beyond the theoretical, beyond the laboratory stage, and into manufacturing reality. Why would anyone commit resources on that scale building a massive factory unless the scaling challenges are perceived to be solvable, or maybe even already solved through advanced biomanufacturing techniques? The apparent speed of institutional execution here, it contrasts sharply with the often decades-long regulatory and developmental cycles we typically see elsewhere, especially in the West.
The factory's very existence, I believe, acts as material evidence that the scientific team and the governing bodies involved believed the leap from the lab bench, where they achieved that 33 months of success, to mass production of individualized treatments is, well, imminent.
The factory confirms intent, yes, and massive investment, and certainly institutional faith. I absolutely agree with that. But intent is not proof of successful scale-up. Large-scale manufacturing of individualized, patient-specific organs is quite possibly the single most difficult logistical challenge in modern medicine. Every single organ produced is bespoke. It requires essentially zero defects, guaranteed sterility, and incredibly timely delivery of complex living biological structures back to the patient. This isn't like stamping out batches of vaccines. We're talking about custom engineering life-saving organs, one by one for millions. The material doesn't really address the intricate quality control or the sheer logistical complexity required to guarantee universal, immediate availability of on-demand kidneys, for instance. So we have to consider the factory as a crucial facility built to test and hopefully solve these scalability challenges, not as confirmation that those challenges are already finished and overcome. If the initial success we saw was based on meticulous, high-touch laboratory work, achieving that same fidelity consistently in a factory setting without potentially catastrophic failure rates, that represents a colossal and as yet unproven hurdle.
But doesn't the very success of the 33-month trial, which presumably was achieved under rigorous laboratory conditions, demonstrate that the process, while complex, is controllable? The factory exists, presumably, to standardize and automate that control. The material strongly implies, I feel, that the core technical process has achieved sufficient robustness to warrant this immediate scaled investment. When you look at it holistically, the definitive clinical success in diabetes, the potentially radical solution to immune rejection, and this immediate commitment to industrial infrastructure, it paints a picture of a scientific program that believes it has cleared the major biomedical obstacles and is moving rapidly into clinical translation and broad application. This looks like hard science that is actively beginning to redefine human health.
I fully agree. The clinical success is definitive for islet cell regeneration in that context, and it's a tremendous scientific triumph. However, precision really remains paramount when we assess the claim of revolution. The confirmed success guarantees potential relief for many diabetics and proves the technique for generating perfectly matched tissue is sound. That's established. Yet, the expansive claims regarding instantaneous, on-demand regeneration of large vascularized organs, and crucially, the systemic reversal of biological aging, these still remain future projections. They seem built on the immense potential of these infant-stage stem cells and the promise of the factory, rather than on confirmed, scaled clinical evidence covering diverse organ systems.
So, to summarize my position, the evidence we have, the perfectly matched, non-rejecting tissue creation potential, and the successful functional regeneration proven by the 33-month diabetic case, these establish a monumental shift in biomedical capability. This confirms the potential, at least, to end reliance on human donors and dramatically alter the trajectory of human health and longevity through cellular renewal. That makes the claims of a near-term revolution, in my view, entirely credible.
And my summary is that while this breakthrough is undeniably profound and has clearly solved a major biological hurdle, the listener really must carefully weigh the secured evidence, which is the 33-month islet cell success, against the truly ambitious, generalized, and perhaps premature projections of immediate impact, the on-demand liver-kidney, the systemic aging reversal made in the material. The value of this technology lies in its immense future potential, absolutely, but its immediate revolutionizing impact across all of human aging remains, I'd argue, speculative until we see proof beyond specialized tissue replacement and confirmation that the Harbin factory can actually deliver that bespoke complexity reliably at scale.
Right. This debate ultimately illuminates that profound tension, doesn't it? The one inherent in any revolutionary scientific discovery, the need to both recognize confirmed success while rigorously scrutinizing the really expansive future implications the material suggests.
Indeed. The complexity here requires us to distinguish very carefully between achieving, perhaps, a masterpiece to certain aspects of human biology and merely finding an extremely elegant and highly effective solution to one, albeit debilitating disease. More research clearly is needed.